Well, thanks very much, Giovanni, for inviting me and also for calling me professor. This was the first time that someone called me professor.
It's like, where it gone?
All right, so I'm here today to present a tool that is based on molecular dynamics, and it's aimed at predictive allosteric binding site in G protein coupled receptors. The code name for the tool is
For me, it was the same before. So you click on the screen, then
on the screen.
No, we already have another one. Is active in another monitor.
Okay. Okay, we just need to find out how to advance the slides. All right. I'll find a way. Okay. So the code name for the tool is MIDAS, which was the acronym of the project that was funded by the European Union. And the aim of my presentation today is to try and convince you that the tool that combines these three key ingredients together can actually be helpful for the slide. Help. Yeah, that can be used for drug discovery purposes. Okay, so I guess everyone is familiar with GPCRs, the largest family of cell surface receptors. They do transmit the signal inside the cell that is brought by a wide variety of extracellular stimuli, and they do this by primarily coupling to G proteins. GPCRs have this conserved architecture made of 7 transmembrane helices that are connected by water-soluble loops and are the target of around 30% of drugs on the market. I have lots of animation, sorry for that. It's gonna be way longer than expected. What most of the drugs have in common is that they do bind its for allosteric binding site, which is the region that hosts the endogenous ligand and is structurally conserved among members of the same family. And this is also one of the leading causes of side effects if the drug is not subtext selective enough. So a strategy to overcome side effects due to lack of subtype selectivity is, of course, to design allosteric modulators. These molecules bind at a distinct site. This is less evolutionary conserved with respect to the allosteric binding site, and they act by altering the efficacy or the affinity of the allosteric ligand. And with respect to that, we can have different types of allosteric modulators, positive allosteric modulators. Everyone knows how this. Positive allosteric modulators that enhance the signaling of the allosteric agonist. And we can have negative allosteric modulators that reduce the signaling of the allosteric agonist, and neutral or silent. Whenever I touch something else happens. Neutral or silent allosteric modulators that bind at the allosteric binding site without altering the efficacy and or the affinity of the allosteric ligand, but they prevent other allosteric modulators from binding at the allosteric binding site. For this very nature, allosteric modulation is subjected to different effects. The first one being probe dependence, which means that an allosteric modulator can be a NAM or a PAM, depending on what is the allosteric agonist in the allosteric binding site. And this obviously complicates translating what we see in the visual experiments to what happens in vivo, because we tend to use synthetic agonists in the experiments, which are more chemically stable with respect to the endogenous agonists, which are instead present when we administer the compound in vivo. Another interesting aspect of allosteric modulation is that when the orthosteric agonist can signal by different pathways, the allosteric modulator can alter one pathway at the expenses of the others, that giving rise to what's called bias signaling. And this can be very useful and exploitable from the therapeutic point of view, especially if one of the pathways is mediating the therapeutic aspect of the compounds and the others are mediating the side effects. This is for example, the aspect that some of the bias agonists of the mu opioid receptor have that can have the analgesic effects without having the side effects normally associated with mu opioid receptor agonism. So there are numerous advantages to try and develop allosteric modulators, which of course, apart from the subtype selectivity, which is expected to be higher with respect to orthosteric ligands, if they lack intrinsic efficacy, they can also have an effect only when and where the orthosteric agonist is acting. So in terms of what happens in the GPCR's world, we only have 3 allosteric modulators that have been approved for clinical use. So the question is why it's been so difficult to develop GPCR allosteric drugs so far. And so until a few years ago, the reason why the rational design of allosteric modulators has been hampered in GPCRs is because we had no idea where the allosteric binding sites were. We started having the first evidence from structural biology in 2013 with the muscarinic M2 receptor in complex with an orthosteric agonist and an extracellular PAM. And we now accumulated over a decade of structural biology methods that show that basically the allosteric binding sites and GPCRs can be virtually anywhere. We have sites on the extracellular and intracellular solvent exposed site, which is somewhat expected because for some of the GPCRs, not all of them, the extracellular site is basically the entry route for the orthosteric agonist from the extracellular medium, and the intracellular side is the region where the receptor couples to the G protein. So it makes sense that modulating altering the aspect of the agonist can be modified, can be modulated by accessing the sites. But surprisingly, many of the X-ray structure and prion structure now that have been solved prove that most of these binding sites are at the interface between the protein and the membrane. So the first structure that came up was the BPTU in complex P2Y1 receptor in 2015. At the time, many scientists thought that could be an exception, that could be maybe an artifact, although it was proved that that was the real binding site. And surprise, surprise, now we know that that is more common than we thought it would be. So a few features that came out from the early analysis and comparison of the X-ray structure that's been solved in complex with and without the allosteric modulators is that some of these pockets are so-called cryptic pockets. We already mentioned today what this cryptic pockets means, but basically the pockets are partially, if not fully, occluded when the allosteric modulator is unbound. So there's no way of knowing that there's a pocket up there if you don't have the compound bound to the pocket. This is the case, for example, of the beta-2 adrenergic receptor that was crystallized. You have X-ray structure of the inactive state of the receptor with an inverse agonist, and without an allosteric modulator, and another structure with the same inverse agonist in the orthosteric binding site and an allosteric modulator binding on the intracellular side. If you superimpose the structure and look at how the allosteric modulator matches and fits in the same pocket in the structure that was solved without the allosteric modulator, you can hopefully appreciate that there's a part of the pocket that is fully collapsed when the allosteric modulator is not bound. So there is some sort of side chain flexibility that happens when the allosteric modulator is bound that cannot be predicted by just looking at a static picture of the receptor without the modulator bound. Another aspect of, especially of the pockets that are the protein membrane interface, is that the membrane shapes and takes part in the definition of the binding site, which is something that I'm talking from the medicinal chemistry perspective, we didn't used to take into account when looking at the definition of what is a druggable pocket. So surprisingly, still today, if you subject the P2Y1 receptor X-ray structure in complex with big BTU to standard web servers that can detect pockets and then can assess the druggability of the pocket. Well, obviously, the pocket where BTPU binds, which is shown in with the magenta sphere, is not listed as a pocket in some of the web servers. But even if the tool is able to detect a tiny pocket there, the pocket will not be listed as a druggable one. But still, there's a drug that is bound to that pocket. So obviously, to be able to predict the location of this binding site, we need tools that are able to take into account side chain flexibility for these cryptic pockets, but also the role of the environment in shaping the pocket at the membrane-protein interface. And obviously, this leads to, talking about computational methods, one computational technique that can take into account this aspect is molecular dynamics. And in particular, there is a MD technique that's been successfully used in the past to detect allosteric binding site in soluble protein, and this is cosolvent MD. And cosolvent MD mirrors an experimental technique whereby a protein crystals are obtained in complex in a mixture with organic cosolvents. And in cosolvent MD, the protein is basically simulated in a box containing a mixture of water molecules and organic cosolvents. The organic cosolvents are chosen so that they can sample different types of interactions that protein and residues can establish with the ligand. And by analyzing the trajectory and looking at the sites where these probes with these cosolvents tend to accumulate and interact favorably with protein residues, this enables to locate the so-called hotspots and map them as allosteric binding sites. So obviously, it would be helpful if we could use this technique straight away for membrane protein. And unfortunately, this is not possible because if you try and run the cosolvent MD in a standard way, as you would do for a soluble protein, the cosolvents will tend to accumulate into the membrane and the system will become quickly unstable. So the idea behind the tool that I'm here to present today was to try and make cosolvent MD possible for membrane protein and in particular GPC nodes. And the way we did this was by replacing the cosolvents with larger fragments that were derived from the analysis of non-allosteric ligands for GPCRs, and by combining them in the form of a cylinder but using a harmonic potential. So we're not actually biasing the simulation, we're just directing the probes away from the water molecules and the membrane. A full description of the protocols and the scripts are available as supplementary information of the accompanying publication. But just to give an overview, they are designed so that you can set up and run the simulation by just running one script. And these cylinders are kept sealed during the calibration so that the particles, the probes, will not start interfering with the system before the actual production is run. And during the production, the cylinder is open towards the protein facing side so that the probe can diffuse into the region of interest. This enables to keep a relatively low concentration of the probes in general, because it enhances the local concentration of the probes in the region of interest, and also prevents the probes from diffusing to distant water layers. Obviously, when you want to sample the binding site at the protein membrane interface, you will need the probes to diffuse into the membrane. So for that, we decided to, instead of using full cylinders, to use small quadrants that the probes will be able to diffuse and start interacting at the protein membrane interface, but that will not compromise the system stability. And as I knew, there were people today presenting serious stuff, actual software, things that work, and very rigorous calculation. This is what's behind the scripts, just me writing down and drawing cylinders for weeks on a whiteboard, and caffeine, and just a tiny amount of mental illness. That's all it's behind these protocols. But despite that, so we validated the protocols retrospectively by selecting 3 case studies. I already presented 2 of them, the challenging beta-2 receptors and the P2Y1 receptor. The 3rd case study was the muscarinic M2 receptor in complex with an allosteric agonist and extracellular PAM. It was the first structure that was available at all as an allosteric GPCR in complex with an allostery modulator. It was the only ternary complex that was available at the time when I was working on this project. We selected these 3 case studies because at the time they sampled different scenarios that were known in the GPCR world at the time, but also because we had X-ray structure of the same receptor solved with and without the allostery modulator that would enable us to double check whether the protocol was able to detect the pocket in either protein configuration. We analyzed, so the probes were derived, as I mentioned, from the analysis, substructural analysis of non-allosteric modulators. And the trajectory were analyzed in 2 different ways. The first one was just to double checking that the probes were sampling the non-allosteric binding site. We did that by measuring the occupancy of the probes and the non-binding sites. We also try and set up a blind analysis to see whether we were able to predict the location of the binding site without having no prior knowledge about it. And we did that by intersecting the density of the probes, so where these probes tend to accumulate during the trajectory, and standard cavity analysis. The results of the retrospective validation were encouraging. Surprisingly, I would say, the protocol was able to detect and open up the allostery binding site, also studied from a protein conformation that did not have the allostery modulator bound, which was something I think extremely satisfactory for us. When we try and set up the blind analysis, obviously for the sites at the extracellular and intracellular side, there was no problem identifying and prioritizing the pockets, because most of these pockets would be we show up in the standard cavity analysis as pseudo druggable pockets. Obviously, the situation was a bit more challenging for the cases at the protein membrane interface, and I reckon that we still need to redefine what we think is druggable in the GPCR world, at least. Well, the tool is able to detect what we already know, which is useful up to a certain point. So we went on and did some prospective validation of the protocol by using it to predict the location of the allostery binding site of a compound that was known to act as a positive allostery modulator for the D2 receptor in the ferritin receptor. And we did that by using the protocol, predicting the location of the binding site, and then suggesting separate immunogenicity experiment to indirectly prove the location of the binding site. And obviously, we did that setting up a collaboration with the pharmacology, which is Professor Peter McCormick. At the time was at QMUL, now it's in Liverpool. This was kind of a challenging case studies because the only extra structure that was available for the D2 receptor at the time was of the receptor in active state in complex with an antagonist, but not the perfect starting point for that type of simulation. So we docked dopamine and the orthosteric binding site, double checked that the docking pose was in agreement with non-experimental NSAR data, derived the fragments from the non-allosteric modulator, and run and set up all the possible simulations with all the cylinders and all the possible placements. And this is just one of the simulation with the extracellular cylinder and the indole probe. This is like the cumulative density of the two different probes for all the simulations. And you can see, binding site are predicted more or less on the extracellular side and the intracellular side. At that time, we decided to prioritize the pocket that was on the extracellular side for two reasons. The first one, because the pocket was predicted to be trackable. And the second reason was because we use an inactive state of the receptor to try and predict the binding pocket of a CAM. So we were not confident that this was a correct depiction of what the active state of the receptor would look like. We dropped the PAM and this pocket, and then suggested residues to be mutated around the ligand to indirectly prove the location of the binding site. Very nicely, we were able to at least indirectly prove the impose on these two residues, tryptophan and the glutamate, in anchoring the positive allostery modulator in place. But once you mutate these two and prevent this hydrogen bond from happening, the compound does not act as a PAM anymore. And what is also interesting is that these results were confirmed by another lab that worked on the D2 receptor and trying to predict the location of the allosteric binding site, and also found the same results with a slightly different allosteric modulator. And later on, the X-ray, the prion structure of the D1 receptor in complex with a positive allosteric modulator on the intracellular side, and two copies of the agonist on the extracellular side, one supposedly acting as a PAM, was located in the same binding site as we predicted in the D2 receptor. So hopefully being able to demonstrate that the protocol, which is nothing less than what we're seeing on the whiteboard, is able to detect fast and efficiently allosteric site and G protein coupled receptors. It really uses a simple trick like an harmonic wall potential, just confining the probes in the region of interest. Simulation will run no longer than 20 milliseconds. You can get results in less than 48 hours for any GPCR. And obviously, in those cases where you have to use it in a prospective way, where you don't have maybe not even a single allostery modulator or ligand for the target that you're interested in, we demonstrated it is possible to borrow some privileged subfragments, substructure from families which are like neighbors in the phylogenetic. And obviously, I have to thank all the people who were involved in this project, the European Union for having Marie Curie, thanks Giovanni and the ALLODD network for inviting me over today. If you'd like to stay in touch, it's a shameless promotion. You found me on LinkedIn and also have an Instagram page where I share the troubles of setting up a computational lab from scratch in Northern Italy. And thanks, thanks very much for the opportunity.
Thanks for the interesting talk. We're running a little bit late, but there's time for more questions. Please.
In your studies, you consider that there is one active state of formation.
Yeah.
Now, in bias agony, several years ago, with Sweden Shine, we proposed that there might be two types of active state. One being stabilized by preferentially by some agonistic line and so on and so forth, and each one having different cytoplasmic ion and so on and so forth. This would be something analogous to what myself have been reporting of the desensitized states, in our case, which may exist in an open and closed conformation. My question is whether for the active state in your situation, you have some evidence for that.
I mean, for the structure that I use for the simulation, I'm not sure I understood the question.
I know that.
So we start from.
If you speak in the microphone.
Oh, sorry. So obviously, if there is a prion structure or X-ray structure of the active state, that would be the ideal starting point if you're looking for a positive allosteric modulator.
But you are not postulating different conformation for resting and active.
No, we use just one conformation for the simulation, depending whether we're looking for a NAM or a PAM. That was the initial assumption.
All right.
Thank you, great talk. My question is that in the post-static validation, you use the risperidone structure into receptor, right?
Yeah.
So we know that risperidone is a bitopic ligand, right? Just using basically two binding sites. One is dopamine site, the other one that is located in the extracellular vesicles. So my question is that you identify this Allostery D2 side for the PAM, but is it really a new side, or is it exactly the same one?
It's not exactly the same one, because it's more far on the back with respect to what was known also for the other muscarinic receptors, for example. But it's nearby, so hopefully that helped during the simulation, having that part of the pocket already open up, but it's not exactly the same location.
Okay, so they are not overlapping with the.
They're not fully overlapping. There's some partial overlap, but not fully overlapping.
Thank you.
Thanks, Antonella. Let's